Good morning, Zoology 2700. Thank you for your brilliant artwork and your addition. These are diatoms from about 100 years ago in a probably a lithograph by Ernst Haeckel. And now they are much more colorful. I like them all. There is a vertebrate, I think, in there, but there's going to be vertebrates throughout the year. We'll accept that. So, the first Friday of the new term. Hooray, because the weekends are different? We shall see. Now, what I'd like to do today is, let's go, and those are amazing drawings. Let's go back, actually. They are amazing drawings, and I will, everybody say cheese. I'm going to screen grab that. Cheese, cheese, do it again, cheese. There we go. And clear all the drawings. They were gorgeous. Well done, you. Admit. So, in lecture 2, back on Wednesday, we spent a lot of time reviewing things that you all remembered very demonstrably well from Biology 1070. We talked about tree thinking and phylogenies, and we labeled different parts of the trees, and we agreed that we would think about monophyletic groups. And what I wanted to do is to put this link up here, and I'll post it in the chat in a second. But this is a review on tree-based thinking that's helpful. That's the link at the bottom. And the tree itself is, of course, the tree, the only illustration in Darwin's Origin of the Species, which is this particular jotting down as he began to express his radical hypothesis that of modification through descent. I wanted, since it's Friday, and since we're all kind of locked down being physically distant and socially connected, I wanted to give you some bit of good news that some of you may have seen earlier in the week, and that is David Attenborough has received his first inoculation of the COVID vaccine in the UK. He's 94, May 8th, I think, 1926 is his birthday. And I feel like we'll eventually be celebrating that. We will in this class anyhow. So today what we're going to do is talk about endosymbiotic theory, about how little things beyond the purview of this course. Remember, most of life, 96 to 99% of named life occurs in this course, but many of the small things do not. And so we're going to talk about how small things became bigger and how metazoans exist as small things, because of course the first part of this course is talking about the protozoa, so protists or protozoans, and this is material that you can read a background about if you're interested in the third chapter of Brusca. Now, before we get too far though, what I'd like to do is to test out continue kind of adding to some of the technologies that we're going to be using to talk back and forth throughout the term. And so this next one is going to be a Zoom poll. So let me start this. So this is a phylogeny. So don't annotate, please, yet. We're going to talk about this phylogeny, which is from the great source of zoological literature, the Wikipedia. And you can see 3 colors on there indicating diaspidids, reptiles, and endothermy. And this is my question for you. The reptile group, how would you describe it? Is it monophyletic, paraphyletic, or polyphyletic? And I'll give you a minute or so to answer. Admit some people into the room. Hello, everybody who's just arriving. We've got a Zoom poll going here based on the screen capture in the background. And I will continue channeling Sarah Harmer as you have your minute. All right, I'm going to end polling, and I'm going to say to you all, well done. That's paraphyletic. So a common ancestor, but not including all of the descendants, paraphyletic. Now, of course, this is an example where you've also got, so the mammalian, the endothermy, that's an example of what we would call a polyphyletic trait. And one of the things that it occurs to me as I kind of look across the evolutionary tree of life and think about applying these terms that describe groupings or placements or thinking about phylogenies is that, a reminder, monophyletic is the key. That's the one that we're really looking for because it gives us a predictive capacity. And once you've excluded monophyly, the variety of poly or paraphyly is not really the main key, unless that's the specific area of evolutionary biology that you wish to dig into the future. Your first goal is to identify monophyletic or non-monophyletic. Paraphyletic groups tend to be taxonomic, so reptiles in this case. Polyphyletic, examples of polyphyly tend to be traits, just like it is here, endothermy. And we'll come back through to some more of these in the future, particularly in the protists. I can think about some polyphyletic things for sure. All right, so thank you for the poll. Put your right hand up, pat yourself on the back. You are poll literate. Well done. So when we're talking today, we're going to be talking today about protozoans. We're going to be talking about eukaryotes. So the presence of a nucleus, karyos, derived from the Greek for kernel or seed. And this is, of course, the scale of metazoan life. I see two things I should have done. A reminder, the chat is in course link, and I will get there in a second. So thank you for that reminder. So the eukaryote, possessing a nucleus, seed, kernel, and this, of course, is bigger and much more complex than the things not included in this class, the typical prokaryotic cell, where we have no true nucleus and essentially the very, very simple line drawing that you've been doing since maybe elementary school, certainly in high school, with a very uncomplicated cell wall and cell membrane and kind of a hodgepodge of no organelles and very little complexity. So the question for us as zoologists that we often come up with is we move from this very simple prokaryotic version of how to be modular life to membrane-bound organelles in this version of a eukaryotic cell. I'm not sure how many of you, maybe you can say in the chat and I'll check there in a second, but I certainly remember having to make a jello mold of a eukaryotic cell where I had to cut up different fruits and vegetables and immerse them in jello, and there was something meant to be the mitochondria. I think it was like stacked apples or something. But anyhow, so my jello mold cell was an abject failure because all of my organelles sank down to the bottom. But we've got so maybe you did, and you can share in this too. So you can appreciate just seeing a line drawing and the fact that in your lab kits, which hopefully you've all ordered by now, there is no jello, and you won't be making a jello mold of a eukaryotic cell. But from an evolutionary perspective, we're interested in thinking about how did this come, where did this come from? And the principal hypothesis about the evolution of complex cells, of eukaryotic cells, is something called the stereoendosymbiotic theory, or SET. There's a figure, 3.52, in your text that talks about this, but this is essentially the hypothesis that in looking at a eukaryotic cell, these membrane-bound organelles contained within a larger and increasingly larger and compact modular cell, is that we're seeing a chimera. That we're seeing the result of, over millions, billions, in fact, years of evolutionary history, the result of simple cells consuming and not completely digesting other moderately less complex cells. So, and the serial aspect suggests that it's happened a couple of times, that the mitochondria was absorbed first, so there was a proto-mitochondria from some aerobic heterotrophic prokaryote that was absorbed, and then we end up with the mitochondria consumed and contained within a larger cellular body. And then for plants and for algae, there was a secondary endosymbiotic event where there was an autotrophic prokaryote, cyanobacteria, something like that, that was absorbed, and that through time becomes the plastid. Now, a chimera, as University of Guelph students, you're familiar with chimeras, this kind of chimeric hypothesis, because your griffin is kind of a chimera, combination of, I don't know, a lion and a chicken, whatever that is. And what I'd like you to do, since you're familiar likely with this hypothesis already, is tell me about the evidence that you know already about this. I'm going to put you into break rooms in a second, so adjust your tie. And 2 to 3 minutes just in those breakout rooms. Open up your mic, your video if you're comfortable. And what I'd like to have you do is share with that group, say, I think it's going to be about 10, how many people do we have in here? 10 people, 5 to 10, say 5 people per group. Open up your mic, chat about, just jot down some of the ideas, and then at the end, I'll send you a note into the ether of your breakout room, and then you can go to this Google Doc that I'll put in the chat as well, where you can enter in some of the evidence that your group has come up with. Clear as mud? Slightly clearer than mud? Okay, I will start the breakout groups, and if there's questions, I will answer them as we go. Breakout rooms. And da-da. Open them up. And as that's happening, I will head over to the chat to see what has happened. Five of you still not in the breakout rooms. That's totally fine. That's your call, but it really interrupts my singing time. Hello again. Welcome back. I hope the feng shui, the arrangement of those breakout rooms was to your liking. I'm hoping that everybody had a good experience in there. And I'd like to thank you for reminding me where I wanted to post that link. Yeah. So here we go. I can see that you're all in the Google Doc now, or many of you are in the Google Doc now, 40 plus of you, and fighting about which cell to enter in. So I'm going to take a look through that as you all rejoin. I think we're mostly back here. And take a look at, yes. So this is great. You're coming up with some great examples here. So evidence for the serial endosymbiotic theory that you may not have known by this name, but that you've seen in the past. The mitochondria has its own DNA. Yep. Bang on. Mitochondria, and that DNA replicates independent of the cell's DNA. Yes. And this is also true for chloroplasts and for, yep, amazing. Double membrane. Yes. Yes. Similar size. Yeah, super duper. Mitochondria similar size to other prokaryotic cells. Circular DNA, reminiscent of other kinds of the arrangement of prokaryotic DNA. Membranes bound around organelles as being vestigial about other earlier independent prokaryotic lineages. Circular DNA compared the size of ribosomes, the double the double phospholipid layer, and room 17, which I guess is code, but it's maybe the room that you were in. But I like it. I will now always remember that mitochondria in the nucleus have different DNA, and I will remember that as room 17. And the ability to have aerobic respiration. That's fantastic. So I'm going to pull that back over here off the screen. Put your hand in the air. Pat yourself on the back. That is a job well done. So let's see, what are some of the other things that we thought about, that I thought about here? And that was, so yes, right off the bat, we start with the same one. Hooray! Mitochondria and chloroplasts have their own DNA that they divide independently. There's actually, I don't know if I saw this one coming up, but there's a different genetic code. The translation from DNA to amino acid takes place via another genetic code within the mitochondria, suggesting an independent evolutionary history. Double membranes that you came up with, the rRNA similarities between bacteria, mitochondria, and chloroplasts. That there's other examples of recent endosymbiotic events in protists that we've actually seen, and then examples of facultative or secondary endosymbiosis. So recent endosymbiotic events in protists or protozoans, what kind of things are we talking about here? So this would be the kind of thing like is shown here. So this is Mesodinium chameleon, which is a protozoan where there is a food vacuole. So when there is phagocytosis, so when kind of the cell membrane, the outer lining opens up and engorges across an autotroph that it's going to eat, so something that's photosynthesizing, you can see here that that food vacuole temporarily acts as a photosynthetic unit. So here from the top in this image, you've got older ingested cell which is just dead now, algal digested. And then there's an intermediate stage here in the center where the algae is almost digested. But in number 1 in the center, the Mesodinium is benefiting from the fact that it delays digestion of that algal cell, and so that algal cell is able to continue photosynthesizing, and the algal cell, the Mesodinium continues to benefit for it. So serial endosymbiotic theory, an important and evidence-ridden hypothesis about the evolution of eukaryotic or metazoan life. And that's, of course, where we're staying now within the rest of the classes, within the metazoans. Now, the origins of life, lots of evidence, but it remains an active area of inquiry. So this is a cover from a relatively recent issue of Philosophical Transactions of the Royal Society in the UK, which is actually the oldest scientific journal. This is a special issue that they had on eukaryotic origin. And there's a link there that I'll put in the chat later on. So if you're interested in the origin of eukaryotes and the foreboding evolutionary puzzle that provides, you can go and read this here. So Phil Trans is an amazing journal. It contains publications by Darwin, by Bacon, and by me. So clearly their quality control is declining through the hundreds of years of their existence. So where did they come from? That's the ongoing evolutionary puzzle and problem. The defining features essentially we're talking about are the nucleus and the cytoskeleton. So let me back off that. Where did multicellular metazoan animals come from? Well, the hypothesis, one of the leading hypotheses, and you can see it in this phylogeny on the left-hand side where all of animals are represented by ants, and the outgroup for all of animals is a single-celled organism called a choanoflagellate. And the hypothesis is that choanoflagellates are the outgroup or the sister taxa to all of the metazoan animals. Now there's a couple of reasons. This is actually choanocytes here. This is looking in, so that the top arrows are looking at two parts. So in the cell A of the right-hand photograph, you're seeing a cell at the bottom and then kind of a collar and then a flagella. And that is the single cell. So it has a long flagellum, microvilli around. Then the long flagellum kind of beats material down into the flagella where they're engorged by the cell. Now, historically, one of the reasons that we like the hypothesis of choanoflagellates being the sister group to all animals is because their incredible similarity morphologically to this cell type in the bottom left-hand side of the image, which is called a choanocyte. And we're going to talk about that in a little bit when we start talking about sponges. But this was intriguing to early morphological zoologists and provided a lot of the original hypotheses about why this group was the sister taxa to all of the metazoa. So single-celled choanoflagellates exist. They're extant species with us. So by talking about the origins, we're not talking about this species that's as old as we are. It's been evolving for the same amount of time. They have a single flagellum with a collar that I mentioned. They're suspension feeders. And choan is from the Greek meaning funnel and describes that microvilli that containing the flagella that allows them to feed and engorge little bits of particles suspended in water. And you can find them either solitarily or as colonial organisms. Now, when did all this happen? Well, we're talking for serial endosymbiosis. Those hypotheses or those events we're talking billions of years ago, 2.7 to 2 billion years ago. So when did the first multicellular metazoan animals appear? Well, 600 million years ago. At around the Cryogean, we had protists and algomats that we can now see as fossils across the world. Often in Australia, it's called stromatolites. The Edicarian fauna, say 600 million years ago, which was jelly-like animal, where the world was principally populated by jelly-like animals, largely sessile creatures, but large fern-shaped things that were suspension feeders likely. And then about 500 to 550 million years ago, 542 to 488 million years ago, for about 50 million years, there was an explosion of diversity. That's called the Cambrian explosion. We're going to talk about that in some detail later on in one of the following lectures. But it's an exciting period of life where where life on Earth went from being jelly, sessile feeding and stalked largely, to mobile, predatory. There were complications in terms of what aspect of symmetry you developed along, and we see an explosion of all of the principal remaining or extant 32 phyla, all occurring in a very small pulse of biodiverse activity during that Cambrian explosion. We also see taxa that appeared there that no longer exist. And so there was just an explosion of ecological complexity in this evolutionary, very dense period. Now we know about this because we know about fossil records. And in fact, as Canadians, we have kind of a lot to say or a lot to be proud of in the fact that within our borders, in national parks, in BC and Alberta, we have protected the perhaps to date the most important Cambrian fossils in what's an area called the Burgess Shale. We'll talk about that in a little bit as well. Now, of course, if you're sitting, and I remember from the maps that you populated on Monday of this week, that many of you are sitting in and around Guelph or in and around Southern Ontario. You're sitting on top of different bits of evolutionary heritage as well. Because until you're up on the Precambrian shield, on the Canadian shield, up in here and above, you're sitting on probably some limestone. So sedimentary limestone that is either Ordovician here in blue. So if you're up towards Ottawa or Eastern Ontario, or even across on Lake Simcoe on the north shore of Lake Ontario, you're sitting on Ordovician rock. So an area that was a shallow, warm sea about 450 to 500 million years ago. If you're down in the southwest of the province, you're in the Devonian period. Your rocks were parts of a shallow, warm tropical sea in the Devonian period, so about 450 to 400 million years ago. And then here in Guelph, we're in a narrow band of Silurian limestone, so 450 to 400 million years old. And these red squares that I've kind of dropped around the map, we're going to come back to these and explore some of the invertebrate fauna that lived here, the marine invertebrate fauna that lived in the spaces where we now live. And we can now look at evidence, fossil evidence in the rock. So in this fossil, there are some crinoids, there's a cephalopod, there's a gastropod, there's probably, if I were to look closely, some little remnants of bryozoan. And this is an Ordovician fossil from Eastern Ontario. And one of the things, I'll give you this link now and you can explore it at your leisure if you're looking to, is I traveled around kind of southwestern Ontario and took gigapans, high-resolution photographs at my feet of stones. And so this is one up in the Ottawa Valley. And you can go, I'll put that link in the chat later on, and you can go and explore some of these fossils. And if you're not getting enough good marine invertebrate time, because probably as we shelter in place, you're not getting a lot of marine time, you can go and revisit these 400 to 500 million year old marine fossils of animals that shared spaces with you in the places where you now live. Okay, so that's a lot of chatting in a while. I want to make sure that I jump over and try and catch up in the chat. And yes, so this is you teaching me that I can't remember what I was supposed to do, and I post the link here. Yes, of course. Hush, Dropbox, I know everybody's working. And yes, there was a couple of questions about recording, and so it's a reminder, we are recording. There's probably 30 of you that aren't able to attend the class synchronously. Yeah, explain how the double membrane evidence is evidence of the serial endosymbiotic theory. And one of your colleagues has answered that just above that. Yeah, it's essentially that you're showing that the prokaryotic cell wall, that it entered the cell and then was consumed but not digested. And so it retained evidence of that original independent cell wall. Oh, amazing. M. Galley is in Alberta, and tons of fossils that are now in the Drumheller Museum. That's amazing. So everybody thinks about Drumheller, well, if they think about it, I used to think about it in terms of large vertebrate fossils, but there's an amazing set of what we were just talking about the other day, cephalopods, ammonite fossils, large curled coiled cell cephalopods that are sister taxa to the extant lineages of, they're nautiloids, there are shelled cephalopods today, but squid and octopus and cuttlefish. So, and yes, that red in that last, when you go back, the red Cambrian line is the Canadian Shield. Pretty cool rocks being in New Brunswick. So amazing. I'm not actually sure in New Brunswick. And you're right, someone up above, so that there is, so they're very old things. So the Appalachian Mountains are up in there as well. But for example, in the Ottawa Valley, we have, where I grew up, there are little pockets of limestone, even though the area is predominantly Precambrian Shield. There are some areas that weren't scraped out by the last glacial maximum. And so we have little pockets of limestone where people try and eke out a living farming, or where young people run around and try and find as many fossils of cephalopods as they can. Okay. So in that final, oh yes, well done on your drawings. I did enjoy them. Let me go back, not that far. And save that. Lovely. Clear all drawings. Perhaps if you have keen ears, you can hear the kindergarten class that's happening in the adjacent room as we multitask, as all of us multitask around the world. Okay. So we'll talk a little bit, so serial endosymbiotic theory, and so basically welcome to the course was welcome to the course, here's some details, and then here's some proselytizing about why it's important to study invertebrates. Then we move through a very quick review of some principal phylogenetic terms and why phylogenetic systematics and the principles or hypotheses of monophyly and Occam's razor allow us to kind of examine the world with a very powerful lens. Serial endosymbiotic theory as a really evidence-laden hypothesis that gets us from prokaryotic to metazoan animals. And now we're going to jump into some of the complex taxa that are contained within the protozoans. This protozoa, Protista here, is a group that used to have its own class long ago here at the University of Guelph, and it likely still ought to. And you could spend a long period of time talking about from a disease perspective, from an agricultural perspective, from just so many different perspectives, different examples of protozoan life. And so we don't have that class any longer, and so it's jammed into here. And so we're going to spend some time celebrating protozoans along with other non-vertebrate containing animals. So protozoans are important for these reasons. Ecologically important, primary producers, consumers, and vital links in food webs all over the world, in marine, freshwater, and terrestrial environments. Humans, you are affected by parasitic protozoans either directly or indirectly. In previous years past, we would have talked about malaria and plasmodium in lab, and I think we still are as an example this year. They reproduce asexually, so there's a replication of chromosomes and then the splitting of that parent into two or more small parts, and that can happen via binary fission or multiple fission or budding. And traditionally, how they moved, whether it was with a flagella, with beating cilia, or with extensions of pseudopods, was really the tools that we used to differentiate groups. And the advent of genetic technologies, as we've been able to look more directly at who's related to whom using DNA, has taught us that those methods of movement, ciliary beating, flagella beating, and pseudopod extensions, are incredibly are traits that have evolved convergently multiple times. And so our understanding of who's related to whom within the large groups of protozoans has been challenged and reshaped a lot in the last 20 years as we've begun to be able to sequence and understand the DNA of the protozoans themselves. Now, there's also still an element of discovery as well. This was in the news a couple of years ago on the East Coast, I think outside of Halifax, where a Dalhousie graduate student went for a walk in a local regional park or conservation area that is pictured on the right-hand side, and they noticed a strange thing. They were kind of down on their hands and knees looking for bits of animal and plant life. They noticed a strange thing. They brought it back to the lab. They took this scanning electron micrograph of it, and what they found was they identified, just in that walk in the woods, a new super kingdom. So kingdom, phylum, class, order, family, genus, species. We're talking about very large taxonomic groups, independent lineage, within which there's a lot of diversity. But until that walk in the woods of that graduate student, we were unaware. So this is just kind of an example that I throw in, as I threw in on Wednesday's class, the Coelenterata, which is the former group that we thought that unified ctenophores and jellyfish. And it was a grad student, a Canadian graduate student, a master's student at the University of Victoria who kind of blew up that hypothesis a couple of decades ago. So emphasizing two reoccurring themes here. One of them is that you can contribute, you are scientists, and you can contribute right away as you take on this rewarding kind of way of exploring the species that we share the world with. And the second is that there's so much to understand about the things that the species and the taxa that we know about, for sure. But there's so many taxa about which we know so little. There's still literally kind of a Wallacean, a Merian, Sibyl, a Darwinian explosion of discovery that can happen because of the tools the new tools that we have. So the Dunn phylogeny, that 32 tip, this is a figure from your text 3.53 that is outside. So the Dunn phylogeny all occurs within, if you look at these kind of Trivial Pursuit triangles, as I date myself again, but these triangles here, so the left-hand side, brown triangle, your Dunn phylogeny, plus all fungus is in here. So in the branching, kind of the fractal nature of branching diagram, so we have in there, right in there, all of that Dunn phylogeny with the 32 animal life. And appended onto that, we have all of fungal life. And then you zoom out one more level even again, and you have all of these lineages that are contracted at the bottom, you can see, by an enormous polytomy, that this is our current understanding of protist relationships. And so within each of these triangles that are spread across this phylogeny, you can, different colored ones, there are large groups whose relationship within and amongst we're still figuring out. And we're going to spend some time, before we jump into the yellow box in the center there called the uniconts, or the multicellular animals. We're going to spend some times outside in groups that maybe with some terms that you've been exposed to before, like the dinoflagellates or the apicomplexans or the ciliates, euglena, euglenoids. So these groups, just to take from this, is that they're very divergent. We don't know which ones are ancestral. It's a large phylogenetic hypothesis containing all of the eukaryotes here, about which we are only one small part. And perhaps this makes you feel frustrated. But I hope not, because what I'd like it to emphasize is just how much we still have to learn and how much you still have to teach and to contribute. So, protists, protozoans, inside of that large phylogeny, small single-celled eukaryotes. The first animals, the protozoans, protozoa, so zoa means animal, proto means first. Some of the first crazed Europeans that came up with microscopes talked about them as animalcules. Animalcules. They can be heterotrophs, they're mobile. There's about 250,000 described species, and they have been evolving on our planet for a very long period of time, 1.4 to 2.7 million years. And so the fossil record is replete with these tiny things, particularly the ones like diatoms, or things that secrete tests, or slightly harder parts that are preserved in the fossil record and transformed into layers and layers of stone. The background reading and some diversifying reading that you can explore here is all contained in Chapter 3 of Brusca. So, there's specialization. That's one of the principal why Why have we seen the evolution of multicellular eukaryotes? And a lot of it has to do with the capacity that are provided for by allowing subcellular specializations. So you can have division of labor. So you can have some part of the cells that are contractile vacuoles that are dealing with excretion. You can have some parts of the cell that are producing cilia and flagella or pseudopods and let you move. You can have some parts of the cell like this oral groove over here. This is a paramecium example that are adaptations towards specialized areas for digestion that are different from where excretion is going to happen. So you have specialization and separation of different tasks. You have extrustomes, which aren't in here, that are about, that can, we're going to see this when we look at paramecium later on, that are involved in defense. And so now when we think about defense and the specialization of labor within a cell, we can think about specializations of lifestyle, things that are predatory. And once you have predators, you need to have defenses against predators. So there, as you build this, you can have either external support, the word test that I mentioned, or a shell, and internal support, like an internal cytoskeleton. So all of these different I guess an example of the extrusomes that I wrote down here for a defense, the trichocysts that are around the edge, those are another example of a defense adaptation. One quick aside that I know maybe I haven't called attention to this yet is that when it says RFB, figure 3.19, if there's anyone out there that's still dealing, maybe you found an old invertebrate text. This is the text we used to use in this class, and there's still lots of them floating around. It's a great text. The drawings are beautiful, and a lot of the description of the biology is beautiful. And the reason we don't use the text anymore is because all the authors ended up dying, and the phylogenetic aspect of it, the evolutionary aspect, stopped being updated. And as we taught alongside that text for several years, there were just so many cases where they would show a phylogeny, and what Sherry and I would have to say is also ignore that, this page, these pages, ignore that, because we no longer think that. Phylogenies are hypotheses, of course, you all know this. And as we add evidence, we change our hypotheses. We adjust and update our hypotheses as we get better and newer evidence. So anytime you see RFB, that's referring to the text. And there is an example of that text that's on, I'm sure you can find one online. So it's a useful text, and it's accepted in its evolutionary hypotheses. And Brusca there is the other figure, of course. So in the last couple of
horrifying monster menace ever conceived.
Easy. In the last couple of minutes, what I'd like to do is to again use the breakout rooms and ask you about protozoans and about what you know about protozoans, and then you will tell me, and we'll either bridge into the next class about what you tell me about protozoans and why are they small. This video is about the blob, and how terrifying the world, what terror we would experience, run, don't walk, if there were large amoeboid animals that could consume or eat you alive. So this is in a cheesy 1950s B-grade horror film. Oh, you're done for, mister. So we don't fear the blob like this. Why not? Why are most protozoans small? So I'm going to put you again into breakout rooms. They're going to be new. So by chance, you might see some of the same people in there. Say hello. And I will post in the chat the re-up the link. Take about a couple of minutes and think about evidence that you could compile that you already know about why protozoans are small, why we don't need to fear the blob. Cool. All right, let me jump back in. See, I see some of you populating. Sarah Harmer is no longer in my head. The current brain worm that I have is if you're happy and you know it. Ah, nice. I like it. Okay. Okay, I see everyone. Everyone's rejoining us from the Zoom land. Thank you. Thank you for helping out and collecting these hypotheses. And what I'd like to do, since we're up right against the end of the lecture, is I'm going to keep collecting those, and we are going to start on Monday from that point. We're going to go through your hypotheses or your collating of the evidence about why protozoans are small, and then we're going to start diving into some of the first taxonomic groups right on Monday. So why are things small? Serial endosymbiosis, that was today. Take care of each other, take care of yourselves over the weekend. Expect kindness and deliver it, and then we'll all get through this together. So have a great weekend. I'm going to turn off for a second, then I'm going to turn it back, my mic and my video, and then I'm going to rejoin. This is, of course, the next hour are my office hours, so if anyone who'd like to stick around to answer or to ask questions, and if not, I'll stay here. I'll turn on my mic and my video and go through some of the remaining messages in the that looks a very sinister cat in the Shah. So well done, and we'll speak to you on Monday or speak to you in a couple of minutes. So